where we left off. You can all see PowerPoint, right? Yeah? Thumbs up if you see the PowerPoint. I got a head nod. Okay, cool. We're going then. All right. So we already talked about sperm cells a little bit. Let's see, produce all throughout the reproductive life. We already talked about that. They're not actually swimming in the testes. They're not swimming in the epididymis. They're not active until they get hit with some of those prostaglandins from the secretions from those seminal vesicle and prostate glands. The ability to fertilize an egg is called capacitation, where they weaken that outer egg membrane. We'll look at that closer when we get to the female reproductive system. Obviously, we'll look at eggs in decent detail, and so we're not going to worry too much about that. So that's where we left off. So here's a picture of a sperm cell. They're a pretty simple thing. If you remember from biology, it's a long time ago, but there was mitosis and meiosis. All right. Mitosis is a cell divides to make two clone cells. It's diploid, so it has two sets of chromosomes. And when it doubles itself and splits in half, it's diploid. It's got two sets of chromosomes. And that's how we're making skin cells, liver cells, muscle cells, all mitosis, right? But then there was also meiosis, where a cell divides and has its DNA doubled and divided. So it's done the same thing, made a clone. But then without doubling its DNA, it divides again and makes 4 cells. And those 4 cells are haploid, which means that they have half the genetic material. So adult humans have 46 chromosomes that occur in 23 pairs. Sperm cells have 23 chromosomes and no pairs. 1 copy of each chromosome, number 1 through number 23. Egg cells have the same exact thing from females through meiosis. Minor differences that we'll talk about in the formation of these, but just kind of bear that in mind. A long time ago, you learned a boring process and you're wondering, why in the world am I learning this? And now here we sit with this. So what do we need to know about sperm cells? The sperm cell is incredibly easy to label. It has a head, it has a midpiece, it has a tail. Honestly, the easiest place to start is probably the tail. So let's start there. The tail is just a flagellum. That's it. You guys know flagella from general biology, I hope. It's a long whip-like tail, works like a propeller to move a cell. That you see them in, bacteria. You can see a couple of them in little protists, little invertebrate things swimming in the freshwater ponds. And that's the flagella in the sperm cell. And it drives that sperm like a propeller does. So that's probably your simplest piece. The next simplest piece would probably be your midpiece. The midpiece is literally just a little cell envelope, if you will, a cell membrane that's full of mitochondria. Obviously, you guys all know from middle school science, high school science, mitochondria is the powerhouse of the cell, right? It has one job and one job only, which is to make ATP. Lots and lots and lots of ATP. And ATP is cellular energy. And that energy is going to be used as the fuel to turn that flagellum. Okay, so if you wanted to, I know my handwriting's just terrible on this, but you could write next to midpiece ATP, because that's what he's doing. He's giving us the energy to fuel that cell and keep that thing swimming. Now, all of this exists as a way to deliver this cell to an egg. That's the goal here. And so the care package, if you will, the thing they're trying to deliver is the head. Now, the head of this sperm cell has a nucleus, and that nucleus is, I don't know if I can write this, I think I can, is 1N. There we go. That's not too bad. So 1N is haploid. It has 23 of dad's chromosomes at random, half of a set, if you will. And the goal is to get that nucleus fused with an egg cell membrane and kind of get through the cell membrane and dissolve and release the nucleus into the egg's cytoplasm. So that nucleus can fuse with the nucleus that mom donated. Now you've got a 2N nucleus, and that is a diploid zygote, brand new baby human, all the information needed to make a baby human, and you're in great shape. And so that's kind of the goal here. Now, to get that job done, you're going to have to get the acrosome utilized. And the acrosome is essentially like a little maybe sheath or covering over the top of the head of this sperm cell. And that thing's job is to hold enzymes that are going to help that sperm get through the egg. And so the egg has a couple of membranes around it. We'll get into that later. But basically, when that sperm cell hits the egg, like this is the wall of the egg, sperm cell hits that thing, the acrosome dissolves and in a little pocket dissolves away the outer membrane of that egg. The sperm can get inside of there, drop the nucleus off, the flagellum and the mid piece, they die, they quit running, they're absorbed by mom, and then the nucleus is there. Like you won. You did it. That was the end. And so that's a structure of a sperm cell. It's not a diagram I have you guys really fill out and label because it's 3 pieces, but I would like you to be familiar with it. So any questions about those structures or what they do? Go once, go twice, 3 times sold. I do not have my phone on me as a clock. So when we get to about 9:28 or so, like wave your hand or give me a thumbs up or something. And because I forgot to give you your plan for the week, we'll just do that at the end. So anyways, there you go. All right, let's see. So spermatogenesis is the production of a sperm cell, right? Genesis is creation. So spermato is the creation of sperm. Pretty simple. And it's not that fancy. I shouldn't say that. I'm probably oversimplifying it. But basically, it's just meiosis. I don't want to write that because my handwriting's so sloppy. We're going to go for cursive. Then I don't have to click. Meiosis. Look, I'm using cursive. It's almost extinct, but not quite. Okay, hooray. Okay, all it is meiosis. It's very simple. It grows just like very similar to the way skin cells grow with a basement membrane. Mitosis, so one cell can stay behind, send that clone up. The clone goes through meiosis, and you're making 4 sperm cells for every 1 primary spermatocyte. Pardon me. I am not going to sit here and split hairs and have you go through and say, well, when is it a primary spermatocyte? When is it spermatogonia? And when is it that? No, that doesn't matter. Okay. The cell at the bottom. All right. Let's pick this guy. Doesn't matter. Okay. Basement membrane. Just like your skin. Good blood supply, good vasculature. That's where the cells are going through mitosis, right? So this cell here is the product of that mitosis, all right? If you know he's a primary spermatocyte, that's neat. I don't really care, but that's cool. And so that cell is going to go through meiosis. So he divides once. You might recall that's called meiosis 1. So he was at 46 chromosomes. He doubled those, and he technically had, 2 sets. He doubles them up, so there's 2 sets here, 2 sets here. And then he goes through what's called meiosis 2. And in meiosis 2, he cuts these 2 cells in half. So he was 1, he doubled and cut in half, then he's 2, he doubles and cuts in half, and now he is 4. And so when you get to these guys, they're called spermatids. They're not quite sperm because they haven't grown flagella, but they're close. They're spermatids, okay? So spermatids are 4 haploid cells. We'll throw a 1N here so you know what's going on. We'll throw a 2. Well, there's a 2 there. 2N here so you know what's going on there. And now we have haploid, I want to say the word gametes. I don't know if they're technically there yet, but let's just go with it. We have haploid gametes that have half of dad's genetic material with a random combination of dad's grandma's chromosomes and dad's grandpa's chromosomes who have made it to any particular sperm, if that kind of makes sense. It's incredibly diverse. Let's just say it that way. Like, insanely diverse. As they mature, and again, we're kind of going into the middle of the little teeny tiny microscopic tubes that twirl inside of the testis. That's called seminiferous tubules. So this down here is like the outside of the tube. This in here is the inside of the tube, and the seminiferous tubule is going to be transporting these guys off to the epididymis. All right. That's the goal, okay? So as these guys develop, that acrosome becomes developed, the mid piece becomes developed, the flagellum gets developed. It's not really paddling yet, if you will, but it's present, it's there. And then that they go into the epididymis for storage. And this guy down here, he just keeps going. He never stops. He just goes and goes and goes. Mitosis, mitosis, mitosis. You go do meiosis. I'm going to stay here and split in a minute. Split again. You go do meiosis. I'm going to stay here and split in a minute. And next thing you know, you're at 280 million sperm, and one of them's got to make it with odds like that, right? So that's how sperm are produced. Meiosis. Pretty straightforward. Questions on meiosis or location? You guys don't have a lot of questions, but it gives me a chance to drink water, so I'm going to ask anyway. All right. Here's a diagram of meiosis. If you don't remember from general biology, which I wouldn't blame you, it's quite a dry concept for the average 16-year-old. Chromosomes are doubled. You split the cell. You do not double them again. You split the cell again. And when you split the cell again, you have these haploid combinations of, let's say, this organism had 2 chromosomes. I don't know of an organism that has 2 chromosomes. That's ridiculous, but sure, whatever, it's fine. So he had 2, made copies, split them. So now he's got a copy of chromosome 1 and a copy of chromosome 2, splits them again. Now he's got 1 copy of chromosome 2, 1 copy of chromosome 1, a different copy of chromosome 1, a different copy of chromosome 2. So essentially, the take-home message here is that every sperm cell is unique. I don't know if it's technically every sperm cell, because with crossing over, I can't do that kind of math. But between friends, I feel like we could say something like 1 in 10,000, 1 in, something like that, 15,000 might be the same, maybe. But your odds of having 2 kiddos that are the exact the same from not being monozygotic twins is incredibly rare because the sperm are so very unique, which is going to be the same exact thing in egg production, by the way, for mom. Okay, crossing over. You might remember this from Gen Bio, you might not. Basically, crossing over is super cool. So since we're talking about sperm, let's just say this is what's going on with dad, okay? So dad has a copy, let's say it's chromosome 6. It doesn't matter, right? This copy of chromosome 6 came from grandma on dad's side. This copy of chromosome 6 came from grandpa on dad's side. So it would stand to reason that when dad goes through meiosis to make sperm cells, you have a chance of getting grandma's chromosome 1 or grandpa's chromosome 1. Kind of makes sense? They split up all the way down. So you might have grandma's chromosome 1, grandpa's chromosome 2, grandma's chromosome 3, grandpa's chromosomes 4 and 5. Now you do that randomly 23 times. So already the genetic diversity you have is mind-bogglingly, ridiculously diverse. What makes it more mind-boggling, ridiculously diverse is this concept right here. When these guys got together during prophase and the chromosomes were all tangled up and we were getting them untangled, you can get a concept called crossing over. And this is super neat. A piece of granddad's chromosome will break off and attach itself to grandma's chromosome. And the corresponding piece of grandma's chromosome will attach to grandpa's chromosome. So if you inherit this set, you're not inheriting grandma's chromosome 6. You are inheriting 70% of grandma's chromosome 6 and 30% of grandpa's chromosome 6. And if you inherit this chromosome, you're getting 70% of grandpa's chromosome 6 and 30% of grandma's chromosome 6. And the even more mind-boggling, ridiculous part is that the same chromosome could potentially cross over multiple times in different locations. So it's like, what the heck? That's so much diversity. So next time somebody says, you got your curly hair from your grandma, you'll know deep down in your heart that nobody knows. I mean, like, who knows where that came from? Like, it's crazy. So that's genetic diversity. Although, honestly, your curly hair could come from your grandma. But holy moly, is it more complicated than that? So those are chromosomes. if you know homologous, or it happens in prophase 1 is all icing on the cake. You should know at a bare minimum that crossing over makes genetic diversity ridiculously high, like mind-boggling, unfathomably diverse. And that's super cool for us, right? Because diverse critters stay alive in hostile environments. So diversity is our friend. Let's see, crossing over. I feel like we explained that reasonably well. I mean, I explained that reasonably well. It's kind of egotistical of me. Do you have questions over crossing over? Going once, going twice? Okay, it must have been explained okay. Or you're asleep. I don't know. So here's some terms. Obviously, I think you should probably already know these, but if you don't, here they are. If sperm cells can't leave the body, then there will be no baby humans made. So some simple things that must occur for sperm cells to leave the body is obviously erection and ejaculation. Write them down if you must. Erection is blood accumulating in corpus cavernosa. Corpus cavernosa fills with blood, and the penis becomes hard. Obviously, that's necessary to get sperm deposited as close to that cervical orifice as possible, because sperm cells are very small and a vagina is a huge space to swim through and navigate when you are a eyeless, noseless, senseless, microscopic cell. So to get sperm cells right up next to the cervical orifice will allow them to basically have the best chance of reaching an egg. You probably already knew that term. The other term's ejaculation, obviously one you probably already knew. It's not like you have to know the difference between ejaculation and emission for your test, okay? But basically, emission is semen is moving into the urethra, which are these smooth muscle contractions that are going through the vas deferens, seminal vesicles, prostate gland, all of these glands, bulbourethra, they're all making these additions. That's called emission. Ejaculation is like right afterwards, and that's when the semen is actually sent out of the urethra. And that's erection, ejaculation. Pretty straightforward terms. Probably nothing too terribly new. I'll give you a second. I see a few people writing, so I'm going to wait. All right. So now we get to the cool part, physiology. Physiology in this chapter is actually interesting. And obviously, it's a little more complicated for the girls, but it's pretty cool. Let's see. All right, hormonal control of male reproductive functions. It's pretty straightforward. Here's what I'd like you to know. There is a region of your brain called the hypothalamus. The hypothalamus is what talks to the pituitary gland. The pituitary gland is often called the master gland because the pituitary gland controls almost everything. And basically, the hypothalamus senses what's going on in the bloodstream and tells the pituitary gland what he needs to do, okay? Now, obviously, the other big player in this is the testes and primarily the interstitial cells that are producing testosterone are the guys we're going to talk about the most. The easiest part to start with this is you need to be able to produce sperm cells, which we've talked a lot about already. Testosterone is a big player in that. But so are luteinizing hormone and follicle stimulating hormone, which we're going to talk about. And then the development and maintenance of sex characteristics, secondary sex characteristics in male. We're talking about thicker skin, larger muscles, facial hair, shoulders broader than the hips. What? I don't know what else. Larynx gets larger, voice gets deeper. That's about it. Yeah, and then I'm sure there's a couple I'm missing, but whatever. The guy's producing sperm, and that's essentially what you need to have happening. So how this all works is not as complicated as you might think. Could you throw in the chat box or let me know what time we're at, though? Because I feel silly not having a clock or a phone.
10:24.
24, 6 minutes. That's perfect. Thank you. We can do this in 4 minutes and then do a plan for the week. All right. So how do you regulate how much testosterone is in the bloodstream? The hypothalamus is the one that's directing this. We're going to talk about that. Don't worry about that. Okay, moving on. Here's the guys you need to know. Hypothalamus can secrete a few different hormones, but this is the one that we're talking about this chapter. It's called GnRH, gonadotropin releasing hormone. And in the world of anatomy names, this name is awesome because it causes the release of gonadotropins. So you could almost like work your way backwards from the testes, and this might honestly be easier. But essentially, what you have is two gonadotropin releasing hormones, which the word gonad literally means testes or ovaries. So the cool thing is there's overlap between males and females. So once you learn this, it's a small jump to get to how the girls are regulating this, which is quite interesting. And so the hypothalamus releases GnRH, gonadotropin releasing hormone. Gonadotropin releasing hormone makes a very short journey to the pituitary gland. Okay, so you want to write that down. Hypothalamus, arrow over GnRH, arrow over pituitary. Okay, hypothalamus, GnRH goes pituitary gland. Tells the pituitary gland. Pituitary gland says, what? He's like, we need more testosterone. Pituitary gland's like, I can do that. Pituitary gland releases two hormones to do that. Luteinizing hormone is called LH. Follicle stimulating hormone is FSH. Follicle stimulating hormone is not turning on the hair follicles in your head to grow hair. Follicle stimulating hormone is turning on follicular or what should we say? I don't know if I like that word. Undifferentiated cells that need to become sperm or egg. Since we're dealing with sperm, we'll say sperm. But for the future reference, it's the same for girls. So LH and FSH. Anterior pituitary takes LH and FSH and puts it in the blood. In the bloodstream, it goes all through your body, but it doesn't bond to muscles, and it doesn't bond to your brain, and it doesn't bond to your liver. It just kind of flows through until it reaches the testes. When it flows through and reaches the testes, LH and FSH are going to bond to interstitial cells and say, hey guys, your time has finally come. Let's make testosterone. Interstitial cells start pumping out testosterone, which allows sperm to develop and allows for the secondary sex characteristics that we just talked about. And so that's how we turn testosterone on, essentially, at puberty, right? But from then until death, there's this long period of time where you're regulating how much testosterone needs to be in a bloodstream. Because once you turn those interstitial cells on, they start pumping out testosterone. Well, you can't just have a never-ending supply of testosterone coming into your body. That's a bad thing. It's a dangerous thing. And so you have to regulate this just like you regulate temperature, just like you regulate the ions in your urine, anything else with homeostasis. And so here's your heavy hitters. LH. If you just know LH and FSH, I am happy with that. If you can't spell luteinizing correctly, you're not breaking my heart. In males, the LH goes to your interstitial cells. The interstitial cells are going to secrete testosterone. Okay? I don't want to write this. It's going to take forever. Testosterone. It's easier if you don't stop and click over and over for every letter. Holy moly. All right. There we go. Okay. In males, LH goes to the gonads, tells the interstitial cells, time to turn on, guys. They turn on testosterone, 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 testosterone. And, voice is deeper, muscles are bigger, guys growing up, puberty, okay? Follicle stimulating hormone gets to the testes, and it is going to stimulate some supporting cells in the seminiferous tubules to be ready to transport the sperm cells and get these guys with their tails moving and that kind of stuff. Do you have to know what LH does and what FSH does separately? No. Just know that those are gonadotropins that are turning on testosterone. I'm more than happy with that, okay? Once FSH, LH, and testosterone levels get high enough, we start the meiosis process and we start making viable sperm cells. And that's about it for male reproductive physiology. It's very simple. I'll show you a really cool flow chart, though, before we're out of time, I hope. Testosterone is the most abundant androgen. There are a few other secondary and tertiary, smaller quantity hormones that will regulate secondary sex characteristics, but testosterone is the heavy hitter for sure. Primarily produced in interstitial cells, already talked about that. There's a very small amount, small amount, made in the adrenal glands on your kidneys. So if you've been curious this time going, well, he said earlier that women produce small amounts of testosterone, but they've got no testicles. So where are those coming from? Adrenal glands and the kidneys. That's the answer. Oh, John, are you waving your hand at me, sir? I'm sorry.
It's 9:30 your time.
It's 9:30. Oh, thank you. Thank you. Shoot. Plan for the week. And we're out of here. Thank you, John. All right. Plan for the week. Today is done. Tomorrow, no Zoom meeting. Excuse me. No Zoom meeting. You guys are going to watch a crash course video on the male reproductive system because I liked your idea of doing the really quick visual stuff after we had covered the majority of the content. And then there will be a quiz on Edmodo, just like last time. That's a homework grade. It's completion. So don't freak out like there's a test tomorrow. No, it's a quiz. It lets me know how much you've retained from 2 days of notes in a crash course video. So watch crash course video, take the quiz. Video's 10 minutes, quiz is 10 questions. It might not take you the full half hour. Sorry. Wednesday, we're coming back for Zoom meeting. And Thursday and Friday, we're doing the same thing. We're going to finish up the guys on Wednesday easily, and we're going to jump into female internal and external anatomy Thursday, hopefully Friday. We'll start physiology.